Hello and welcome all of you. We are into the general pharmacology sections and we have already covered pharmacokinetics. So let's jump to pharmacodynamics. We know what pharmacokinetic is now, what body does to the drug. And there are different aspects of pharmacokinetics, that is absorptions, distributions, metabolism and excretions. So that was all about pharmacokinetics, and now we are going into one more section, that is what drug does to the body. How drug produces its effects, what are positive effects, what are negative effects, how they producing it. So these all things, these questions would be answered in this section, and that is pharmacodynamics. So it mainly deals with what drug does to the body. So it's basically a study of drug effects. Now, how this drug produces its action, that is also called as mechanism of action, and you'll hear this term throughout these pharmacology lectures, that what is the mechanism of action, how drug produces its action. There are different ways. Some drugs, absorbs the UV rays and produces its action. Some are having absorptive property, prevents absorptions. Some producing osmotic activity. Drugs act by radioactivity as well, oxidizing property, neutralizing property. For an example, antacids, which is used in acidic environment. But majority of these drug actions created with interactions with another molecule that is made up of proteins. So major drug action which is being carried out by interactions with target biomolecules, and those target biomolecules are proteins. Now we'll see briefly what this target protein may be. This target protein may be, first, it may be enzymes. And we already know what is the role of enzyme. An enzyme which helps a substrate to get converted and to create a product. That is the role of any enzymes. So our drug might interfere with this process. So it may produce no product, or it may produce a non-functional products. So this is how a drug may interact with protein, and this protein might be enzymes. So interfering with function of enzyme is one way of interactions of drug. These are some of the examples. For an examples, endogenous substrate is acetylcholine, and which is metabolized by an enzyme cholinesterase. So our drug physostigmine and neostigmine interacts or interferes with the function of this cholinesterase or inhibits this cholinesterase. Same way, some catecholamines metabolized by MAO, that is monoamine oxidase enzymes. So with this enzyme, the drugs which interferes is moclobemide. And this moclobemide is mainly used as an antidepressive agents. Levodopa metabolism occurs with dopa decarboxylase, and interference occurs with the help of few of the drugs, that is carbidopa and benzirazides, that we will see in CVS section. So these are some of the examples you can remember where drug interferes with target protein, and that protein is enzyme. This protein may be ion channels, right? We know the ion channels. The function of ion channel is basically transfer of the ions across the cell membrane. So there are some substances or endogenous substances which acts as an opener of this channel. So this channel gets open, and some ions gets inside the cell and some ions goes outside the cells. So this process is being carried out. But drugs may interfere with these functions, that they may block this channel or they may modulate these channels and produce its different actions. For an example, quinidine, which is mainly blocking the sodium channels present in myocardium. Nifedipine is a calcium channel blocker and mainly blocks the voltage sensitive calcium channels. Nicorandil, Nicorandil opens up ATP sensitive potassium channel. So these are some drugs which we use in cardiovascular system to modify different actions by blocking those or opening those ion channels. Drug may interfere with protein, which in the form of transporters. We already seen this transport system across the membrane, where substrate is being carried out substrate transport is being carried out with the help of transporters. So drug may interfere with this transport function so that substrate no longer gets on the other side of a membrane, and which may interfere with the function and produce its effect. So these are some of the examples. In the synapse, this is presynaptic neuron and this is postsynaptic neuron, where some of the molecules gets reuptaken in the presynaptic neurons, and this reuptake process is being carried out by a few of the transporters. For an example, norepinephrine transporters. This norepinephrine transporter transports the norepinephrine in the presynaptic neuron, but few drugs like desipramine and cocaine blocks this reuptake process by inhibiting those transporters. Same way, some serotonergic transporter or serotonin transporters, which transports the serotonin across the membrane, and this transport function is inhibited by drugs like fluoxetine, we mainly use in depression. Some dopamine transporters are also there, which transports the dopamine across the neuron, and which may be inhibited by drugs like amphetamine. So these are some of the examples where drugs inhibit those transporters. And lastly, drugs may interfere with this protein in a form of receptors. Now, what are these receptors? Receptors, we already know, they are like a lock and key mechanisms where they are having an acceptor site where endogenous molecules or any molecules, or I would say a ligand, binds to them and produces its action through different signaling process. Drug may act as an agonist, which may bind and produces the response, or drug may interfere with the response by in the form of antagonist that we will see in later half. So drugs may interfere or modulate the action of this receptor and produces its response. So these are the four target biomolecules where drugs may act. The drugs may act on enzymes, drugs may act on ion channels, drugs may act on transporters, and drugs majorly affects the receptors. So majority of functions of interactions with protein is being carried out with the help of those receptors. So we'll see obviously in brief or in detail, I would say, about receptors. Now, first question arises, what are they? What are receptors? So receptors are basically a macromolecule or a binding site. So they are a kind of binding site which is obviously made up of proteins majorly. Where it is located? So it is located, that might be located on cell surface or transmembrane or inside the cell. So it may be located anywhere, and it serves two functions. It serves to recognize the signal molecule. So where a signal molecule or a drug attaches to it. So first thing is the attachment, and second, it produces the response. So receptor having two actions, serves to recognize signal molecule or drug, so that gets attached, and second, it initiates the response. So that is a receptor. It's a macromolecule. Again, I'm telling you, it's located everywhere. It may be inside the cell, outside the cell, on the cell membrane or transmembrane, and it serves two functions. It produces attachment site, and second, initiates a response. Now, what is ligand on the other aspect? Ligand is any molecule which attaches selectively to the particular receptor or a site. So here only one aspect is there, attachment aspect. Right? So ligand is basically more associated with attachment, a molecule which attaches to the particular receptor or a site. Now here we are having two different terminologies that we need to understand. One is affinity and second is efficacy. Now what is affinity? Affinity means ability of drug to bind with its receptor. So first aspect we discussed. So ability of a drug or a ligand to bind with the receptor, that is called as affinity. So affinity of a drug, that means that drug attaches to the receptor site. But what about efficacy? Efficacy is capacity to induce a functional change in the receptor, which is called as intrinsic activity. So first was affinity is ability to bind with the receptor site, and second, what is efficacy? To produces functional changes and produces intrinsic activity and to initiate the response, that is called as efficacy. So these are two terminologies we need to now understand: affinity, ability of bind to the receptor, and second, efficacy, to produce intrinsic activity in receptors. So depending upon these two properties, we have categorized those, agents, or endogenous agents, or I would say endogenous or exterior agents. So depending upon these two properties, drug is being divided into different types. Which kind of that drug is, whether this drug is full agonist, antagonist, inverse agonist, or partial agonist. So we'll talk about all of these things. Now let's see what's in this figure. This figure is showing two things here. It's showing a response. Here the response gradually increases, and here it's showing drug concentration, right? So if you increase the drug concentration, what happens to response? So let's see. In full agonist, what happens here? Here the response is maximum. So maximum response we have achieved, you can say, that is 100%. So maximum response here is of agonist. Half of the maximum or the submaximal response is of partial agonist. No response, you can say, or neutral response, or the no response is in antagonist, and negative response is in inverse agonist. So that is, that is been described in this figure. Now let's see what they are saying. So what is agonist? Agonist is an agent which activates a receptor and produces full effect, right? So agonist activates the receptor and produces full effect. For an example, say +2, right? So that is agonist. So agonist activates the receptor, is drug, receptor, and effect, I would say +2 for an example. What are inverse agonists? Inverse agonist activates the receptor but produces response in opposite direction. So drug which acts on receptors and produces opposite effect. So I would say, for an example, in this example, this is inverse agonist, so it produces -2 response. So that is inverse agonist. So inverse agonist produces opposite response. What about antagonists? Antagonists are the agents which prevents the action of agonist on a receptor, and it's not having any own response. So here the drug and the receptor would be having no effect, right? So I would say a neutral, zero response. If agonist having +2, if inverse agonist having -2, this would be having 0 because it produces no response. It just prevents the action of agonist. And what about partial agonist? It activates the receptors and produces submaximal effect. So now if we need to conclude this, full agonist, I would say full agonist or the agonist produces +2 response, partial agonist produces +1 response, antagonist produces 0 or neutral response, and inverse agonist produces -2 response. And these are the different definitions of these agonists. Now also we need to understand about two-state receptor model. One more theory about this agonist, partial agonist is the state of receptors. And they've been saying that receptors are in equilibrium state in resting phase. Equilibrium between activated receptors and inhibited receptors. So receptors are basically in two states, in active form and one in inactive form, and both these forms are generally in equilibrium. So this is a resting equilibrium status between active form of receptors and inactive form of receptors. Now what happens with the help of agonist? With the help of agonist, activated receptors numbers or response increases in comparison to inactivated, so it produces the full response. What happens with antagonist? Antagonist activates both activated and inactivated, so eventually there won't be any response or the zero response. What about partial agonist? Again, partial agonist tilts the state more in the favor of activated receptor rather than inactivated receptor, so it produces partial response. And lastly, inverse agonists, they shift the momentum with inactivated receptors, so inactivated receptors state increases and it produces opposite response. So this is a two-state receptor model by which the theory of activated receptor and inactivated receptor is established, and the different types of agonists and antagonists are explained. Apart from that, the receptors are of different types, and these are some of the short questions that can be asked in examinations. Like for an example, what are orphan receptors? Well, there is a very interesting thing. Orphan receptor means those receptors for which there is no endogenous ligand or mediators present. No endogenous ligands or mediated presence in the body. Those are called as orphan receptors. So for them, the agonists are those which only administered from outside. So that is why they are called as orphan receptors. So endogenously in the body, there are no agonists present of that receptors. Some of the examples are benzodiazepine receptors. There are some benzodiazepine receptors present in central nervous systems where there is no endogenous ligand. No endogenous ligand attaches to these receptors. But if you administer some exogenous drug, for an example benzodiazepines, those benzodiazepines will only act on these receptors. So these receptors are mainly made for exogenously administered drugs, and these are called as orphan receptors. Second question is, what are spare receptors? The theory established of spare receptors after we noticed that if a agonist is being given, there is a full response, right? There is a full response, we've already seen. But if we now check the status of receptors, so in some cases only less than 1% of receptors are being occupied. Still, even after the full response, we have 99% receptors left. So what are those 99% of receptors? These are spare receptors. Still a lot of receptors are left. Still we are getting full response. So that is how the spare receptors concept came into the picture. So many full agonists produces maximum response even occupying less than 1% of available receptors. There are still lot of receptors left, but we are getting maximum response with less than 1% of occupancy. And that is why those receptors which exist are called as spare receptors. What are silent receptors? They are more of a ligand. Silent receptors means those are the sites or receptors where a drug binds, but no pharmacological response is elicited. For an example, plasma proteins. We have seen there are plasma proteins in the blood which attach some of the drugs. Drugs bind to those plasma proteins, and they only act as a reservoir. They don't produce any kind of response or pharmacological response in a body. So they are only acting as a site of binding. and these are called as silent receptors. So these are few of the short questions that can be asked in your examinations. Second thing or terminologies you need to remember are drug action and drug effect. These both of things are different. What is drug action? When a drug binds to the receptor, it produces conformational change in the receptors or prevents the conformational change. So that drug might be agonist or antagonist, but in short, what is drug action is a drug is binding to the receptor site and produces conformational changes in receptors or prevents the conformational changes in receptors. That is drug action. While what is drug effect is ultimate change in the biological functions due to that. So because of that, if any biological function gets changed or series of reaction or response happens, that is called as drug effect. So now you see both of these two terminologies. So drug action is ability of a drug to bind to the receptors and producing conformational changes or prevent the conformational changes. So this drug action is limited to receptors actions. And what about drug effect is eventual or ultimate response of change in biological function, that is drug effect because of the consequences of drug action. So these are two different things you need to keep in mind. Now, how the receptors are classified? If the receptor produces a particular pharmacological action, so depending upon the pharmacological criteria, which pharmacological action it produces, on that basis they are classified one way. Second, where they are distributed. So depending upon the organ of distributions, they can be classified. Their ligand binding. What is their ligand? What is their endogenous substances? Okay. So depending upon those endogenous substances or their ligand, those receptors are classified. Transducer mechanisms. What is their way of response? How these receptors gets activated and how they produce its response. So on that mechanism, they are classified. That is transducer pathway. And the last is the latest one, the molecular cloning. That few of the newer receptor subtypes been developed and they have been identified. Now many and many subtypes been identified. So depending upon the subtypes, the receptor is been classified. So there are different ways of classification of these receptors, but we'll see or we'll discuss mostly the transducer mechanisms of types of receptors. So different types of receptors depending upon the transducer mechanism.